You are listening audio paper series with Yasir, and today's guest is Dr. Thomson from Quadram Institute, Norwich, UK, to discuss one of his publications. A whole genome assay identifies 4 principal gene functions that confer tolerance of meropenem stress upon E. coli. It is published in Frontiers of Antibiotics. Welcome, Nick.
Hello.
Why did you study E. coli susceptibility to meropenem?
Well, meropenem is a beta-lactam antibiotic, which means that it's a drug that kills bacteria by preventing the cell wall from forming properly, which kind of weakens the structure of the cell and makes it more likely to lyse. So beta-lactam antibiotics generally are used very widely in medicine, but there's a problem recently with accumulation of antibiotic resistance, and especially within the beta-lactam class of antibiotics. And so understanding the mechanisms behind how the antibiotic works and also how cells combat the effects of the antibiotic is important for discovering new drugs or using the drugs which we have in an appropriate way.
Okay. So how did you study these susceptibility mechanisms?
Well, what we did is we decided, first of all, that we were interested in studying the drug at high concentrations, but also at low concentrations. So we know the concentration of meropenem that usually kills E. coli. And so we grew the cells at that concentration, but also at double the concentration and also below the concentration that kills the cells. So that meant that we were looking both at below the critical concentration for killing the cells. We were looking at cells which are stressed by the drug, but not actually killed by it. When we look above the critical concentration, then that allows us to identify genes or functions of the cells which are important for tolerating higher concentrations and might give a clue to how this resistance is evolved.
Okay, so you studied susceptibility mechanisms at different concentrations of meropenem.
Yeah, so after we'd exposed the cells to different concentrations of meropenem, then we used a technique called Tridus Express, which allows us to measure the effects of every gene in the genome of E. coli simultaneously under the conditions of meropenem concentrations.
Cool. Okay, so what are the 4 principal gene functions that confer tolerance to meropenem that you have mentioned?
Overall, we identified about 300 genes, which is actually a surprisingly large number. But we found that the majority of them can be grouped into one of 4 functions. So the first 2 functions were not particularly surprising. They were for cell division and for synthesis and maintenance of the cell wall and the cell membrane. So these are basically the mechanisms which we know are targeted by the drug. The other 2 were transcription regulation and ATP metabolism. And these ones were slightly more surprising because there's less known in the literature about how meropenem relates to those 2 functions.
Okay, so you did identify the mechanisms that are already reported, and you also identified 2 new, relatively new mechanisms.
Exactly, yeah. So that shows the power of Tridus Express because you're looking at all of the genome at the same time.
Okay, so there were some already known mechanisms and some novel mechanisms are found in this study. How can we use this understanding to improve susceptibility to meropenem?
So because we now have an overall understanding of all of the gene functions that are involved in this tolerance to meropenem, it means, first of all, we have a better understanding of how the cells react to the drug and also how the drug actually interacts with the cells to kill the cells. So that allows us to look for drugs that have perhaps a synergistic mechanism together with meropenem. So one In the paper, we identified one drug called cefsilodin, which targets one of the genes which we found makes cells more susceptible to meropenem. And so that means that if we use both drugs in combination, then we have a bigger effect than if we use either drug on its own.
Okay.
And so knowing things like this allows us to put together different combinations of drugs, and also gives us clues as to how we might be able to design new drugs that will combat the effects of antibiotic resistance.
So we have potentially identified a synergistic mechanism as well by studying this data.
Yes, exactly.
Lovely. Thank you very much, Nick. That's very informative paper that you have shared with us today.